No, sorry to start it. Yeah. Well, it's the early organizational stages. Can you hear me?
Okay, can you hear me now?
Now I can. You must have been
Okay.
muted before.
Right, right. And then I hit something else on my screen. How are you this morning, Dr. Adachi?
Thank you, Jacqueline. You sound like just like me with this Zoom technology. Meaning
Oh,
dated.
so, I have, do you hear an echo or no?
Nothing that
Okay, good. Because
bothersome.
we have a Zoom meeting that starts at 7 and I just logged out of there and I wasn't sure if I had closed it because sometimes we get an echo. Now, I have your slides loaded on my computer.
But it's probably better if because I need to move them around.
Correct.
So
Correct.
shall I share screen?
Yes, why don't we have you share? You have to have the presentation up on your desktop, but then if you share your screen, then we can make sure that we can see your slides.
Let me try that.
Good morning.
Good morning, Catherine.
How are you?
Good.
Just 1 second. Can you see it?
No, I'm seeing a Google Calendar.
Calendar.
Can you see it?
No.
We can see your screen, but not the slides. Dr. Adachi, you might want to close the, some of those apps you have open.
I'm not sure what apps I have open.
Okay.
But usually this works.
You know what? Open the PowerPoint presentation.
I did.
Oh, well, we're not seeing it.
Right. Hold on a second.
Okay.
You still don't see it, huh?
No.
Then we have a problem. I'm not sure because I have done this before many times. Not sure what the issue is. So if you were to put them up, I'm not sure how it will work because I need to advance the slides. It's quite elaborate.
Yes, I went through your slides.
So I really need to be able to control it.
Okay. What are you seeing on your screen right now?
I see both of you.
Okay, so if you
Do you have two monitors, Dr. Adachi, or one monitor?
I do have two, but
Okay.
I thought I put the presentation on the right one.
Maybe we can switch it off to the other monitor. Maybe perhaps we might be able to see it.
Right. At this point, I don't see my share screen though. So let me see if I can somehow. I may, given my technical skills, I may have to start from scratch. Is that okay with you?
yes. So Catherine, does Dr. Adachi have can he control it if I open the slides?
No, whoever's sharing the slides has control over the screen. Dr.
Okay.
Adachi won't be able to control the screen.
Okay.
But Dr. Adachi, I see I see Facebook and on the top tabs. If
Yes.
you look on top, maybe if you can close some of those tabs. we might-
No, that's
no?
that's not the issue. I really don't.
What's showing on below those tabs is that we're seeing that you have Google Calendar open. And
Yeah, but when I share, usually all of that disappears and you just see the slides. And I don't know why this didn't happen here.
can
Can
you open the presentation?
Yeah, it's open.
Oh, it's open. Okay.
Yeah. And in fact, I brought it to the screen that I thought I shared, and didn't work. You're
Okay.
saying that you don't see it. Here it is. I see it on the right screen, presumably, but you don't.
No.
So something's not working.
Dr. Adachi, do you see the Outlook calendar? Maybe you can minimize that and we're able to see the slides.
I can minimize that, but I'm not sure that will help you.
Yeah, why don't we have you minimize that and then see?
I just did.
Oh, okay. I see your cursor on the minimal.
Can you see the slides now?
No,
Yeah.
we're still seeing the calendar.
Well, the calendar was minimized, so I'm not sure why you see it. So we're having a problem. I wish it wasn't so, but I'm afraid that's the case.
I'll tell you what, why don't you
I'll start.
close your, why don't you exit share screen and let me see if I can pull it up here. And
So you're saying I should stop share?
stop share and let's see what's going on. Okay, so you see the three of us
Yes,
on your screen now.
I do.
Okay, in the background, so at the bottom of my screen, I have PowerPoint. And if I open that, can you see your slides now?
No, I cannot.
No, Dr. Lachlan.
Ah,
What's this? Are you
okay.
sharing your screen, Dr. Lachlan?
Oh, well, let me minimize this and then, all right, so share screen. Okay.
No. All right, now you're sharing your screen. Okay,
Okay,
that's the
so
first. We can see the slide now.
you can.
Yes, in presenters mode.
I know you have a lot of reveals. Can you get a view on your computer screen, Dr. Adachi, where you see the presentation in a small box?
Well, I see the presentation. It's not in a small box, but I see it, but I cannot move it. And
No.
because you probably will have to control it.
Okay, well, I stopped sharing.
Yeah,
So
good.
click on it in the small box, and if you have the slide sort view, and then share your screen, and then we'll see if we can work this out.
So could you repeat that? What?
Okay, so you have it on your screen. You can see the presentation.
I can put it on, yeah. I don't have it now.
Okay, so just have it on your, can you put it on your desktop?
Yes, it's on my desktop.
Okay, so can you open it? We'll go away, open it, and then go to screen share.
Open it and then go to screen share.
Yeah, open it first.
Um, yeah. Not sure I can do that because that covers up my screen share, my share.
Yeah,
screen.
no, let it cover up your screen share. That's okay. And then it should appear at the top of the screen. So
Or the
open
bottom.
the presentation.
Yeah, I don't see now the share screen though.
It should be,
It should be on the bottom of your screen or on the top, Dr. Adachi.
Well, it is, but once I bring the slides over, I don't see it.
Okay. You're not seeing share the tab for sharing the screen on the bottom or anywhere?
No, once I move it over, I can't see the share screen.
So click on the share screen.
Okay, I did. And I'll share the slides again. Hopefully it will be the right. Hopefully it will be the right screen. Huh. Yeah, I see the problem, I think. I don't have now a view of my second screen. I wish our IT people were here, but I didn't think it would be a problem.
do you just want to use one screen and open the presentation on the one screen?
Well, I may have to. Let's try that.
Oh, there we go.
There we go. I can see.
We can see it now.
Okay, so then if you just go down to, yes, put it in the slide presentation mode. I see you
Slideshow.
just
There you go, that one.
slideshow, yeah.
Okay,
Click that.
perfect.
And then if you want to maximize it.
Towards the
Dr.
right,
Lachlan?
towards
Dr.
the right, top right, Dr. Adachi.
I'm sorry, one at a time, please.
Go towards the top to the right, and the middle icon, you can make it bigger.
I'm sorry, could you repeat that?
Towards the right, top of the slide, right, the middle icon, keep going to the right, towards the right, there you go, the middle icon, there you go.
This one?
Yes, yes,
Saying click on it?
yes.
That's, no, yeah. So somehow
Okay.
we're not on the right screen, of course, but at least you're seeing something.
Yes, what we're seeing is the speaker view. And
It's not
So
what
if
we
you go
meant.
to display setting, hey, it's Mark. Thank you so much for joining us, Dr. Adachi. Go to display settings and you'll see swap presenter and speaker view. It's up at the upper left-hand corner. It says display settings.
Yes, I see that.
Go there and you'll see swap. Go ahead, drop down.
Yeah.
Swap presenter and slideshow. You're right there. There you go. Perfect. Thank you.
Who was this Deus ex machina?
Please, I'm Mark Einstein. I'm the chair of the department. Welcome. So we appreciate you joining us.
Einstein, you could not be Deus ex machina.
I guess you could call me the AV team, okay?
Dr. Einstein, that's why he's much more experienced than some of us in terms of the getting the graphics up here.
That was like the voice of Zeus from the Pantheon.
Okay. So welcome to OBGYN Grand Rounds. Our Grand Round speaker this morning is Dr. Elliot Adachi. Dr. Adachi is the former Dean of Medicine and Biological Sciences at Brown University, where he's a tenured professor. Dr. Adachi is an academic physician executive. He's a graduate of Tel Aviv University School of Medicine and the Harvard School of Public Health program in healthcare management. Dr. Adachi did his residency training in OBGYN at Tufts University, and then went on to a fellowship in reproductive endocrinology and infertility at Johns Hopkins University with the Joneses, and then on to the University of California at San Diego, where he was under the leadership of Dr. Aaron Shui and Dr. Sam Yen. Dr. Adachi has been elected to the National Academy of Medicine of the National Academy of Sciences. He served on many of their consensus committees. He's been a mentor to over 50 postdoctoral trainees and fellows, and he is the author or co-author of over 500 peer-reviewed publications, 120 book chapters, and he's been editor or co-editor of 16 books, as well as serving as a member of an editorial board of multiple journals. He's the recipient of continuous National Institute of Health funding for 20 years, and he served on numerous study sections at the NIH. Among his many honors, he is the recipient of the inaugural Lifetime Achievement Award from the American Society of Reproductive Medicine, and he has also been awarded a Certificate of Special Congressional Recognition. I could talk forever about Dr. Adachi's accomplishments, but we'll go on to his presentation, Eggs and Sperm from a Buccal Smear. Is this the end of infertility? Dr. Adachi.
Thank you very much for this very kind introduction, and it's a pleasure to visit with you again. I remember visiting in the days of Dr. Gerson Weiss, for example. All of that, of course, is long gone. I wish I could be there in person, but that wasn't to be this time around. The subject matter is futuristic, you might say, but it is in many ways closer than we realize. And I will, of course, expand on that and try to convince you of that. And just in general, let me say that when we speak about stem cell-derived lineages, they are hardly limited to the reproductive arena. There are investigators in the neuroscience field who are trying to create amacrine neurons for the retina, yet others who are seeking to create dopaminergic neurons for the striatum, for example, in the context of curing Parkinson's disease. There are those who are actively pursuing the generation of trophoblast precursors and motoneurons, which could revolutionize the situation for multiple individuals who are paralyzed because of a disruption of the spine, who could, in principle, walk again. And then, of course, there are the gametes, which are the subject matter of today's discussion. But I wanted to make the point that this is hardly an isolated case of stem cell-derived lineages. I also thought it's useful to point out to you the priorities that were set by the Nuffield Council on Bioethics in the UK, which is the premier bioethical institute of the United Kingdom, which under human reproduction proposes to first tackle, in terms of the horizon for bioethics, in vitro derived gametes. That says something. They placed in vitro derived gametes at the top of the list, to be followed by time limits on maintaining embryos in culture, womb replacements, preconception screening, whole genome sequencing of embryos and fetuses, genome editing of gametes and embryos, and egg freezing. All of which is to say that there are other organizations the world over that are recognizing that in vitro gametogenesis is approaching fast, and that it may indeed influence in a dramatic fashion the way we practice IVF and reproductive endocrinology in general. This is, of course, a disruptive technology. Whether or not it's achievable in the human cannot be said at this time, but we are at least part of the way towards achieving this goal. Whether it's going to be a short or long timeline, again, are impossible to say, but this is an active area of pursuit and will likely materialize before too long. When we speak about these types of futuristic projects, it is always good to recall the work of Aldous Huxley, which was best represented by his 1932 book, Brave New World, wherein he predicted that eggs will be produced via ex vivo ovarian cultures, that embryos will be generated via in vitro fertilization, and that newborns will be delivered via ex vivo ectogenesis. Of these three predictions, and there were more, he was definitely right on when it comes to embryos being generated via in vitro fertilization. This is, of course, our reality today. But in vitro gametogenesis was not on the screen of Mr. Huxley at the time. That is something he could not envision because the developments that make it possible occurred substantially later. You may recall that in chapter 1 of Brave New World, the director of the hatcheries, so-called, says, 1 egg, 1 embryo, 1 adult equals normality, making 96 human beings grow where only 1 grew before progress. As you will see, that is, of course, exactly where we are with in vitro gametogenesis. We will be able to have 96 well plates replete with gametes, be they sperm or eggs. We could select by a variety of criteria. We could edit those gametes. Much could be accomplished by having those gametes available to us in vitro in a dish, as predicted in a way by Mr. Huxley. For the longest of time, the notion of in vitro gametogenesis was deemed infeasible because of the so-called soma germline barrier hypothesis, which was formulated by August Weismann, who was a professor of zoology at the University of Freiburg, and who articulated his theory in his book, The Germplasm, A Theory of Heredity. The essence of it was that the germline, of course, gives rise to the soma, which thereby is immortal. but that the soma never gives rise to the germline and therefore is mortal. Recent technology, of course, changed all of this and in a way reversed these dogmas, which at this point you could argue are no longer valid. And that was because of the advent of somatic nuclear reprogramming, the idea that you could take a differentiated somatic cell, convert it by different means, which we will discuss, to a pluripotent stem cell, and then guide that pluripotent stem cell towards a gametogenic fate. The first technology that made this possible was known as somatic cell nuclear transfer, or SCNT in short, and came about in 1958, courtesy of John Garden, who was a professor at Cambridge University in the UK, who published a seminal paper in Nature titled Sexually Mature Individuals of Xenopus laevis from the Transplantation of Single Somatic Nuclei. In a word, the work was done in frogs. Starting with a somatic karyoplast or a nucleus, which was transferred into an oocytic cytoplast, that is to say, an oocyte that was enucleated, he created what you could think of as a zygote, which proceeded to develop into a blastocyst from which one could derive embryonic stem cells. In 2006, a new technology was introduced for the generation of stem cells that was known as the induction technology, and it was brought about by Shinya Yamanaka from Kyoto University, who published a seminal paper titled Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors. What Dr. Yamanaka did, he started with a somatic cell, let's say a skin cell. He transfected it with select transcription factors that he identified. And that generated what is now known as lowercase I PSCs, or inducible pluripotent stem cells. Both individuals, Dr. Garden and Dr. Yamanaka, understandably were awarded the 2012 Nobel Prize in Physiology and Medicine. In vitro gametogenesis simply takes off on these principles. You start with a somatic cell, let's say a skin cell. You convert it either to an embryonic or an inducible pluripotent stem cell, and then you guide it by a variety of stimuli in vitro towards a gametogenic fate. One would have to ask oneself, what are the prerequisites for taking an undifferentiated stem cell and guiding it towards any particular cell type, in this case, a gamete? Well, you would have to know the program, if you will. You would have to know the signaling mechanisms, what I call here the germ cell specification pathway. What cues do we need to provide the stem cell to differentiate into a gamete? And what sequence should be used to apply those cues, and by which timing? And ultimately, are there other players required, such as surrounding cells, which are sometimes referred to as the niche? And as you, of course, all know, oocytes do not develop in a vacuum. They are surrounded by granulosa cells and by theca cells. And by the same token, spermatogonia are surrounded by Sertoli cells. And so there is a niche requirement, it would seem, in the context of gametogenesis. The modest beginning of this field can probably be traced back to 2003, when a paper saw press in Science, first authored by last authored by Hans Schöller, who at the time was at the University of Pennsylvania, but now is at the Max Planck Institute for Molecular Biomedicine in Germany. What he was able to do is to derive so-called oocytes from mouse embryonic stem cells, an image of which is shown on the right lower hand side of the slide. But this was early on and really not much differentiation or fine-tuning could be assigned to this putative oocyte. That same year, Professor Noche from Japan reported on the pages of the Proceedings of the National Academy of Sciences the same feat, as did Dr. George Daly from Harvard Medical School and Renee Rejo-Pera from UCSF. So at the time, some early indications were that we could potentially start with a somatic cell, convert it to a stem cell, in this case, embryonic stem cell, and from there produce, hopefully, germ cells. Many papers followed suit in the years thereafter. This is a series of examples of papers where germ cell specification pathway of the rodent was defined, and a series of papers wherein the germ cell specification pathway of the human was defined. In a word, we were making slow but sure progress in understanding what it would take to take a stem cell towards a gametogenic fate. So as we reflect back on the state of the art in 2015, we would have to say that the germ cell specification pathway is not conserved between the rodent and the human, which foretold the fact that it will be difficult, or more difficult probably, or require separate efforts to make this happen in the human. And as you will see, that panned out. We know how to do it in the rodent. We have yet to figure it out in the human. And we also realized, I think at that time, that the genetic networks involved between the rodent and the human are quite distinct as well. Another way of looking at the state of the art in 2015 is to say that we figured out, if you will, the extragonadal component of gametogenesis, meaning we could identify embryonic stem cells or inducible pluripotent stem cells and even convert them to what is known as primordial germ cell-like cell. However, we were not able to go beyond that point to the intragonadal component of gametogenesis and end up with haploid, postmeiotic, mature gametes. In a way, we figured out the extragonadal component. We have yet to figure out the intragonadal component. So the final frontier is really, particularly in the human, in vitro gametogenesis that goes all the way. In 2016, Katsuhiko Hayashi from Kyushu University in Japan published on the pages of Nature a paper titled Reconstitution in Vitro of the Entire Cycle of the Mouse Female Germline. In a word, he figured it out, but for the mouse germline rather than the human germline. He started with embryonic stem cells or induced pluripotent stem cells, led them to so-called primordial germ cell-like cells, and then combined them with gonadal somatic cells, which led to the creation of early primary oocytes, eventually M2 oocytes, which upon fertilization and transfer to a surrogate mouse gave rise, first of all, gave rise to a blastocyst, which could be transferred to a surrogate mouse, which gave rise to pups. In a way, then, the title of the paper was true to form in that Professor Hayashi did, in fact, start with stem cells and end up with pups. The same was attempted in the human, but not as successfully. In this case, Mitinori Saito from Kyoto University published on the pages of Science a paper wherein he started with human primordial germ cell-like cell induction, combined it with xenogeneic ovarian stem cells because we don't have, at this point, engineered, if you will, ovarian somatic cells. And he was able to take the process all the way to the oogonial stage. But that's as far as he could get. We could not get to the point of female determination and meiotic initiation. In a word, we can make it happen in a mouse, but we cannot make it happen in the human as yet. And this is as far back as 2018. To give you a sense of the importance that is being attached to this field, I am displaying here a recent paper that was published in Science magazine, essentially a review on mammalian in vitro gametogenesis by the two principles we just alluded to. That tells you something about the significance the scientific field attaches to the process of in vitro gametogenesis. The same is true for the New England Journal of Medicine, which saw fit this year to publish a paper on the creation of oocytes from stem cells under the series known as Clinical Implications of Basic Research. All this goes to show that two of the leading journals that we all recognize chose to display the state of the art because they, like others, perceive, and I think correctly, that this will happen, and it's just a matter of time. To further illustrate this point, I can share with you that the National Academy of Medicine is planning a 2022 workshop in Washington, D.C. that is, among others, supported by the American Society for Reproductive Medicine, but also by other foundations. The whole idea is to, for once, be ahead of the game rather than follow the New York Times headlines. In other words, anticipate the development rather than chase it after it happened. So where we are today in terms of the missing links for terminal human gametogenesis, we need additional knowledge of the germ cell specification pathway, the generation of gonadosomatic cells or the stimulation of their input, the so-called niche element. And we need to be sure that we complete the replication of meiosis in a faithful fashion. This is very well specified and spelled out. Back in 2014, Marianne Handel from the Jackson Lab defined so-called gold standards for what it would mean to have meiotically competent in vitro derived germ cells. Those cells would have to have the correct nuclear DNA content, the normal chromosome content and organization, appropriate synapsis and recombination, and viable euploid offspring generated. So before we bless, let's say, the human in vitro derived gamete, once that feat has been accomplished, we would have to meet the gold standards set by Marianne Handel. These would be the minimum requirements for us to be convinced that what we have is what we think we have. This, as you, I'm sure, have imagined by now, has significant scientific and translational promise if it happens. From a scientific point of view, it will provide an inexhaustible supply of germ cells. It will provide for functional non-mutant analysis, which advances the knowledge of functionality. It would allow for the study of germ cell, somatic cell interactions, about which, by the way, we don't really know a whole lot, even though we have studied granulosa cells and stem and theca cells for the longest of time. But this can also have significant clinical implications. You could reverse, in principle, germ cell failure. You could engage in autologous gamete replacement rather than use donor gametes. You could gene edit those gametes or replaced gametes. And you could even use those gametes for donor mitochondrial replacement therapy, rather than call on another woman who would have to be stimulated, her eggs retrieved, etc. So possibilities that in many ways are really endless. You can imagine Turner patients being able to conceive with their own oocytes, unthinkable, or other cases of ovarian failure that can be addressed, perhaps, by this technology. This will also have a significant effect on IVF as we know it. It will eliminate the need in stimulation and egg retrieval, which currently occupies a significant amount of our time and effort. It might do away with gamete donation, perhaps with the exception of age dependence. We don't really know whether in vitro derived gametes from an individual in her 40s, say, would be appropriate and workable. And that would still have to be, I think, established. I think predictions are that this will convert IVF essentially to a laboratory procedure, because you will not have to stimulate and retrieve. I mean, you will still have to engage in endometrial preparation and transfer of the embryo, but the type of activity we currently engage in is likely to be substantially modified, you could say even curtailed. Whether or not all of this will improve the affordability of IVF and thus the access to it is a big question mark. We've been there before. The promise is often there. The reality is not necessarily always materializing. All of this, of course, will have to be carefully regulated, and that will fall onto the FDA. This is FDA jurisdiction, specifically the Center for Biologics Evaluation and Research, or CBER for short, and that will likely take the in vitro derived gametes into a category they refer to as human cells, tissues, and products, or HCT/Ps for short. But this technology could not be brought into the clinic absent the blessing of the FDA. There are also a significant number of ethical considerations, most of which we will not have time to get into. On the one hand, this is a welcome development because it is compatible with the principle of Hippocratic beneficence. It will be helpful to patients, and therefore we need to embrace it and welcome it, ethically speaking. By the same token, it could relieve infertility. Yet again, a beneficent action that we ought to embrace. It will, however, bring around echoes of the commodification of reproduction. You'll get a better sense of this in a slide or two from now. Simply because of the scale, we are going from 10 to 12 oocytes, let's say, retrieved per cycle, to essentially unlimited number of oocytes in 96 well dishes that could be stacked up a mile high in an incubator. As you will see, there'll be novel and you might say perhaps less than the salutable notions of parenthood. And there'll be the so-called enhancement overhang out there, because once we have those gametes at our disposal, they could potentially be edited and enhanced in ways that, at the very least, deserve very careful consideration. Some enhancements most of us can agree on. For example, if we could enhance humanity to counteract the COVID-19 virus, in other words, if we could have it in our genes, I don't think many of us would argue with that proposition. But of course, that is an obvious example. Many others are far less clear. And I go back to what I said about the magnitude of the phenomenon. We are going to be producing essentially unlimited numbers of gametes from individuals, culture them in 96 well plates, and we'll have to somehow deal with this massive influx of gametes, which we have not, as a rule, been accustomed to. And that will require selection, which in itself raises a number of ethical issues. Should we place limits on scale? Should we place limits on choice? And how should we dispose of these products? This almost certainly would benefit from a serious deliberative effort by an appropriate body in every nation, including in the United States. The National Academy of Medicine would be one appropriate body that could explore this new phenomenon, ideally before it happens, and develop a set of guidelines that are clear-cut, well thought out, defined by experts, that we can then follow once this actually comes to pass. There used to be a presidential commission for the study of bioethical issues. For some reason, and I'm not sure I understand why, the Biden administration did not reestablish this commission. The Trump administration definitely did not do so. But the expectation was that the Biden administration would, and that did not happen. Not clear why. And then we have the Hastings Center in upstate New York, which is the premier bioethical center in the United States, which could certainly take on the deliberative effort involved in the matter of artificial gametes. And as is always the case, there will be legal implications. You now have an overabundance of oocytes and sperm sitting in dishes or in freezers, and all kinds of possible unreal scenarios could be envisioned. Just to illustrate, although this is not very likely to happen or ever going to happen, imagine the possibility of unwitting, non-consensual parenthood. Imagine a scenario of you having a drink in a restaurant, leaving the cup after you're done, somebody picking up the cup, scraping some of your cells off the cup, converting those to gametes, and before you know it, using those to fertilize the opposite gamete, implanting those, and without your knowing, you may be the father or the mother of an offspring you never knew about or may never know about. I'm only using this example, which could well be deemed as unlikely, to illustrate the complexity of the phenomena that could transpire and the legal implications thereof. Apart and distinct from leading committees that take on an issue like this, there is now ever-growing emphasis on the idea of public deliberation or participatory public engagement. The idea that you and me would also be part of the discussion. I don't mean you and me as professionals, but you and me as citizens of this nation. When you look at nations like Denmark and Holland and other European democracies, you begin to realize that public deliberation is part of the governing process. Virtually every critical decision that is ultimately going to be voted on in parliament or executed is brought up before the people, and public deliberation of one kind or another is transpiring. We really did not appreciate that concept, and perhaps the first time this was brought to public attention was in 1975 in the wake of the so-called Asilomar Conference on Recombinant DNA, which was an important conference, but involved scientists only. No public was invited. So experts only, public excluded. This, I think, triggered the sequence of public participation in the United States. This began with a comment by the late Senator Edward Kennedy, who in May of 1975, in a speech at the Harvard School of Public Health, said, "When science develops techniques that fundamentally change society, society has the right to determine how the technique is to be used, whether it should be developed in the first place, and if so, under what constraints." Other senators, such as Senator Javits, also commented on this issue in a subsequent Virginia conference. A 1995 report of the National Academy of Medicine on essentially social and ethical decision-making in biomedicine called for the same principle. We need public participation when it comes to leading social and ethical decisions in biomedicine. In 2008, the National Academy of Medicine dedicated a whole report to public participation, in this case in environmental assessment and decision-making. But a relevant statement made there was that public participation is intrinsic to democratic governance. Something that, of course, our European colleagues figured out long ago, but which was important to articulate in our context. The leading proponent, I would say today, of public participation is Professor Sheila Jasanoff from Harvard, who in her book, Science and Public Reason in 2012, said that the issue is no longer whether the public should have a say in technical decisions, but how. And that is, I think, an important trend to recognize that in the United States, at least, may be slow in coming, but there are advocates for such. Whether or not we will ever reach that corresponding promised land remains to be seen. I'm showing this only to illustrate how well the British are doing it. They, under the leadership of the Royal Society, conducted public dialogue and opinion related to genetic technologies, which, as you know, are exploding. But they wanted to know what the public thought and how they should proceed with that insight in mind. And then a report was issued, in this case in March of 2018, which captures what the public had to say. And that informed not only scientific bodies, but also Parliament, which in the United Kingdom often rules on various scientific and breakthrough developments, for example, mitochondrial replacement therapy. The Nuffield Council for Bioethics, which we started with, also published an important contribution on genome editing in human reproduction. But germane to the point that we are discussing right now, it said, establishment of a separate body or commission, independent of government, which would produce an understanding of public interests through promotion of public debate, is in fact an important principle. And that is what we were trying to articulate earlier. And that is what the Nuffield Council on Bioethics so elegantly and so succinctly made clear. Which brings us to where we started, to the agenda, if you will, of the Nuffield Council on Bioethics, what's on the horizon for bioethics, and the fact that in vitro derived gametes is really what they consider to be the next frontier, or the next frontier that we need to be ready for and therefore tackle today. So with that, perhaps we can close for now. I leave you with the statement, stay tuned, because that's really where we are. It's could happen tomorrow. You might open the New York Times and there it is. Or it might take much longer than we project, in which case there may be a National Academy report issued by then, and more of the science would be available to assess. But for now, we're somewhat up in the air, even though the collective sense is that this is inevitable, that this will happen, and that we better get ready. So thank you very much for the opportunity. And should there be any questions, of course, I'll be happy to answer those.
Thank you, Dr. Adachi. Are there any questions from the audience? I have a question. It's a little off topic, but where do we stand with the CRISPR technology and germline editing?
Right. Well, and that's relevant, I think, to what we were talking today, although it's in some other respects, it's a separate topic. But yes, as I think was just said, it's been a decade now since a gene editing enzyme known as CRISPR was discovered, for which a Nobel Prize award was already provided, and which was being considered, is being considered for the possibility of editing. Up until now, most of the discussion has been about editing embryos with an eye towards correcting monogenic diseases to begin with. As you know, there is a tsunami of monogenic diseases that proceeded uninterrupted for as long as there is humanity, because we had no way of interrupting the transmission. This may well be the first time in human evolution where we can actually intervene and prevent the birth of children, or the inevitable birth of children who are affected by this or that monogenic disorder. There were some early efforts to edit human embryos that were published in some of the best journals, but the state of the art is such that we are nowhere near knowing how to do this safely and effectively. In fact, if you edit an embryo with today's technology, all kinds of new pathology is being generated, including, and I emphasize, an outright loss of a chromosome or two, all of which obviously we don't want. In a word, the technology is still too crude to be applied for clinical practice. And that was, in fact, the recommendation made by a joint commission of both the National Academy of Medicine and the Royal Society of Medicine back in 2020. The latest paper on this issue was published, the latest papers in this issue were published in Cell from the group of Professor Egley at Columbia University, and in PNAS, the Proceedings of the National Academy of Sciences, by the group of Kathy Nayakan from the Crick Institute in the United Kingdom, both of which in this highly credible, high impact journals made it plain that the technology is nowhere near ready for prime time. And so, promising, intriguing as it all is, today it is simply infeasible and inadvisable and essentially verboten because contraindicated, because it's simply not working. Now, to bring us back to the subject matter of today, it is entirely possible, not certain, but entirely possible that if we came to the point where we had in vitro derived gametes in a dish, in multiple dishes, we could do the editing at the gamete level in the dish or in multiple dishes and select through various selection mechanisms the correct clone, if you will, or the correct gamete, and use that gamete for fertilization. This is all theoretical speculation on my part, but one of the potential advantages of in vitro derived gametes is that we will have lots of them in multiple dishes, that we could edit them in vitro, which is easier than editing an embryo, which you almost have to do in conjunction with ICSI. And so it's possible that in vitro gametogenesis might ultimately be the vehicle for germline editing. Time will tell. We are just not there yet.
Thank you. Are there any additional questions for Dr. Adachi? No? If there are no questions, then the code for CME is 11335. And I want to thank Dr. Adachi for this wonderful presentation and his time. He's a true scholar, and it was wonderful to hear where this field is going. I see Dr. Einstein has in the chat, which I'm not sure you can see, Dr. Adachi, but as well as Dr. Borelli, that they all want to thank you very much for this presentation. Good to see you.
Good to see you all.
We'll see you in person one of
I
these
certainly
days.
hope so. Keep thinking about me.
Right, we will. And
Okay.
thank you very much. I see Dr. McGovern is on. I'm not sure. if you can
Yes,
see him.
Nice to see you, Dr. Darcy.
good to see you, for heaven's sake. It's been way too long,
Yes,
way too long.
Well, maybe in a year or so, we'll be having some in-person
Yeah, let's work on that.
presentations and exchanges.
Let's work on that.
Yes,
Yeah, no, I always enjoyed visiting. As I said, this goes back a long time when Dr. Weiss was the chair, I think. So yeah, I'd be pleased to do that. So let's keep that in mind.
great.
Okay,
Thank you very much.
thank you. Bye-bye.
Bye-bye.